Perry and Brianna for that introduction. I'm really happy to be here today. Even before I started working with Dr. Perry in 2011, I was actually a master's student here at George Washington University. Not in the School of Public Health, but I was in the School of Engineering and Applied Sciences. I did my master's degree in environmental engineering management. So I was here in, I think that's when that was. I think it was 2007 to 2008 I did my master's program here. And it was actually during this time where I was in my kind of first year doing coursework, and I had done a bachelor's degree in math and environmental toxicology. So at the time when I was applying for graduate programs, I was really kind of struggling to figure out how to merge the two together because I really enjoyed both the environmental tox and the math courses that I had taken. So I figured engineering was the perfect fit to merge those two. But it took about 2 months in an engineering program to realize, oh, I am not an engineer. I think an engineer takes a very specific way of thinking and a way that your brain works, and my brain does not work in that way. So it was during that time where I actually started taking some courses outside of the CEAS school, and I ended up in Dr. David Michael's class where he was teaching, I forget what it was called, but it was an introduction to environmental health or environmental risk assessment, something along those lines. And I just fell in love with that course, and I realized public health was the place for me. And so that's when I applied for doctoral programs. I don't think we had a PhD in environmental health here. I think that's relatively new, right? I saw on the website, I was really excited. I was like, this was here 6 years ago. Definitely applied to that. But that didn't work out for me.
Story.
Yeah, it would have been a totally different life story. But that's okay. It ended up working out okay because just up the Beltway, there's Johns Hopkins University. I ended up going there and getting my PhD in environmental health policy, where I worked with Dr. Thomas Burke. And that was a fantastic experience, which kind of led me into the fields where I'm working today. So I'm really excited to be here to talk about some of my systematic review work, and then also thinking about kind of the future of systematic review and thinking about mechanistic data as well. And I'll talk about why that's important. So my outline for today, I'm going to talk a little bit about the impetus for systematic review and why it was important, and then give a brief overview of systematic review methodology and how the framework is structured, and then thinking about how we can apply this to mechanistic or in vitro data. And if you're not familiar with that type of data, I'll talk a little bit about what that is and why it's important to us. So this was basically the impetus for systematic review in a way. It was people were identifying how we made decisions in environmental health and identifying many limitations that existed in the way that we made decisions about how to regulate environmental chemicals. And there were certain authoritative bodies like the National Academies of Sciences that would review what EPA would do in terms of either risk assessment or their IRIS program where they do chemical assessments. And they would list out all of these things that were challenges. So it wasn't transparent, it wasn't really clear how they were coming to their decisions, it wasn't clear how they were selecting studies to base their decisions. And obviously these have wide-scale impacts, right? If EPA decides we're going to regulate a chemical and this is going to be the reference dose by which we say things are safe, things are not safe, that basically dictates how the world assesses or regulates chemicals, right? Because the US will regulate it in that way, and then other countries will look at what the US has done and they'll adopt that as well. But there's all these limitations with the process and how it's done. And so some people wanted to keep things the same. Some people said, it's fine, we've been doing it this way for 30 years. And another group of people said, no, we really need to start thinking about how to make this process better. And so that's how systematic review came about. So most of you, if you've been in the environmental health field or taken some environmental health classes, you already kind of know the depth of statistics that demonstrate how environmental health in the United States and in the world has very serious problems, right? So autism is one example where we have many autism cases being diagnosed, more so than can be attributed to changes in genetics or just increase in diagnoses. We see more severe types of health outcomes, particularly in children. A lot of my work focuses on reproductive and developmental health, so I focus a lot on diseases in either developing fetuses or very young children. And so thinking about things like childhood cancer, where we've seen a 63% increase in number of cases of diagnosed children, kids with cancer from 1975 to 2014. So we're seeing these huge increases. Similarly, if you are looking at neurodevelopmental disorders, things like autism, but also things like ADHD, we see a consistent rise over time with diagnosis. And again, this is more than can be attributed to just increases in diagnosis. In other words, more attention being raised to this issue and clinicians kind of looking for and diagnosing it more. We're actually seeing an increase in the amount or the number of children with this disease. And just overall, in general, we're seeing troubling trends in kind of life expectancy and disease, as well as mortality rates. So here's showing a graph from 1990 to 2008, thinking about different racial, ethnic groups, as well as being broken down by those with education and those without. So thinking about the interplay of socioeconomic factors as well. In particular, for white women without a degree, we can see that the life expectancy has decreased, I think, by about 5 years over time, between 1990 to 2008. So we see a lot of trends in environmental health that are pointing to kind of a grim story, right? There's a lot of issues. And a lot of these we just don't know a whole lot about. And I think you can see this exemplified very well in the news and the media, where oftentimes you'll see the press pick up on one particular scientific story, and they'll kind of create a splashy headline, and then kind of summarize the study, but obviously they kind of leave a lot out from the study, right? A lot of times they kind of jump to implications when maybe the study authors didn't really feel that strongly that there was a causal relationship between the two things, but the news media kind of makes it sound like that, right? We see that a lot, and this is why there's a lot of confusion, I think, both in the media as well as within the general population. One example here is there's kind of two things where we seem to see stories in the news like basically every week, and a lot of times they say opposite things. So those two things are coffee and red wine. It seems like every month or so there's a new story about why red wine is good for you, why red wine is bad for you, and then same thing with coffee, like beneficial health effects and the detrimental health effects. But overall, we know that there is a problem in environmental health, and a lot of it might have to do with this. So the U.S. chemical production from 1945 to 2007 has increased by about 15-fold. So right now there's about 83,000 different chemicals that have been registered for use within the United States. That's a lot of chemicals. So more than 80,000 different types of chemicals that can be in the food that you eat, the packaging where your food is being kept, in your homes, in your schools, in your workplace, in the furniture that you're sitting in right now. There's a whole host of chemicals that you could be exposed to. And even though we have 87,000 chemicals that are registered for use, we really only know health information about very few of these. So this is what we call tip of the iceberg. At that tip of the iceberg, we have about 200 chemicals where we have at least some health information about them. These are things that you see scientists publishing papers about, saying, BPA is linked with low birth weights, or a chemical is a carcinogen, or it's being linked with ADHD, or it's linked with autism. There's only about 200 chemicals where we have studies that demonstrate health effects. The rest of these, now I have to think, 79,800 chemicals are kind of below the surface. We basically have very little health information about the rest of these chemicals, but they're still being manufactured and used in the United States. So this is the problem that we're trying to deal with. And again, EPA is the main government agency that addresses this issue for environmental chemicals. And we have to think about, how do you take all of the information that is out there for at least those 200 chemicals, and then synthesize it in a way where you can clearly justify making a decision. Should we act? Should we ban it? Should we set a regulatory standard? What should that regulatory standard be set at? All of these decisions are really challenging. There's several reasons why they're challenging. One of the main reasons is because there's a lot of studies. So if you're looking up in this upper left-hand corner, any of you who have tried to do like a background lit review and you're putting in like arsenic into PubMed, you get like 30,000 papers, right? A lot of them may not be relevant to what you're looking at, but you'll still find hundreds and thousands of peer-reviewed scientific journals that publish thousands of articles on the same chemical, and you have to kind of sift through all that information. That takes a long time to do. Even if you're a scientist at the EPA who's doing this full time, that's what you're paid to do, that takes a long time to do. It's an issue of person, I almost said manpower, but I guess I should say person power. The other problem is these studies will be conducted in very different species. So you can have studies that are done in your very typical mouse and rat species, but then you can also have studies that are conducted in zebrafish, or C. elegans, or chickens. You also have epidemiology studies that are conducted in humans. Now, how do you take all of that information, like if something's toxic in a zebrafish, and it's toxic, but it shows another health effect in humans, and then in mice it shows something completely different. How do you interpret all of that information together and make inferences ultimately for humans and human health? That's a really challenging thing to do. This bottom graph, or figure, shows that in addition to different animal species, including humans, you have different experimental designs that you might use. And if you've taken an epidemiology course, some of these might look familiar, thinking about case control studies, cohort studies, case series, case reports. These are all done in humans. And then you might have animal research as well. And then at the very top here, kind of what we might consider the gold standard would be things that we typically see for things like pharmaceuticals. We see randomized controlled trials in humans, where you can randomize an exposure and then see what the health effects are. This is pretty rare for environmental health, right? We can't randomize two groups of people and say, I'm going to expose you to some air pollution, and you, I will expose to no air pollution, and I'm just going to wait and see what happens. There's a lot of ethical reasons, there's a lot of study design reasons where you actually can't do that in environmental health. So we have all of these different types of epi studies and animal research, and the quality of each will vary depending on how it's been designed and how it's been implemented. So that's another challenge. The last challenge is something that we encounter all the time in academia, where you might have two individuals who read the exact same study, and they come to totally different conclusions about it. One expert might say, well, this study didn't do this, and so I don't think it's a valid study, and I don't really believe in the results. And the other investigator might say, no, but they controlled for it this way, so I think this is actually the best study I've ever seen, and I put a lot of faith in their results. You can have that argument all day. So all of this combined means that when we're searching for arsenic studies, and we get all of these studies in different journals, in different species, of different quality, and then we interpret it differently, that means it's really hard to do a chemical assessment where we figure out, is this harmful? And then if so, whether we need to act, and what levels we need to set regulatory standards at. So this is a hard thing to do in general, and what made it even harder was this is kind of how we did it in environmental health before we had systematic reviews. So this is what we call narrative reviews. It's basically an expert or a few experts who get together in a room, they find all the studies that they think are relevant, and then they summarize the findings. And usually they summarize the findings in a way that kind of looks like this, which basically sums up to there are some positive studies, there are some negative studies, and we need to do more research because it's unclear which one wins out. So there's a lot of issues with this, right? There's issues with, you don't see it in that paragraph, but usually when you're reading a narrative expert-based review, there's always issues with transparency. You're never quite sure, well, how did the study investigators find those 20 different studies when you do a search and you can find 3,000 studies in PubMed? How do they narrow it down to those final sets of studies? Reproducibility is an issue too, because if you don't have transparency, basically if you don't tell someone, I did this, 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 and this, they can't reproduce it on their own, right? So if somebody just went and tried to do a search in PubMed, they might not get the same studies, they might not get the same results, and that's a problem. Because in environmental health, when you're making decisions for millions of people, you want to make sure that an independent third party could get the exact same results that you got. This was a very resource-intensive process. So a lot of times for EPA, their chemical assessments can take up to 10 years to do. Benzene is one example, where I think they started an assessment in 1992, and they published it in 2002. So there's literally people at the EPA who can work full-time on one chemical assessment for a decade of their career on one chemical assessment before they get it out. So it's so resource intensive that obviously in 2002, if we said, okay, between 1999 and 2002, sorry, 1992 and 2002, there's probably going to be more studies published, right? We need to update that search and redo it and figure out what the new literature says. You might spend another decade trying to do that. don't know if this is bad. It says I've stopped sharing video. Do I have to worry about that? Okay. Then the other thing is it's very resource intensive, right? So you have to make a pretty significant resource and investments in terms of like time and money spent on systematic reviews. So, on narrative reviews. So if you're doing a narrative review, it's going to require a lot of person power, and it's going to require a lot of money. So over that 10-year period, the EPA could spend a couple $1 million just funding that one review on that one assessment. And remember, we have 80,000 chemicals that are out there being used in the world, right? So if we're trying to do this for every single chemical, that would be very challenging. And then the last two things. So first, it's really hard to justify your policy decisions. If your whole process is not transparent and it's not reproducible, the NAS has done critiques of EPA assessments before and basically said, I have no idea how you came to your final decisions. That means it's going to be really hard to justify, oh, we need to ban this chemical, because somebody can't follow the steps that you took in your process to come to that decision. And then lastly, with narrative expert-based reviews, it doesn't really indicate where the science is flawed or where we would need more research specifically. So it doesn't have that kind of feedback loop where it informs, if you wanted to conduct a new study, where would it be best to focus in order to improve the overall scientific literature base? In other words, where are the knowledge gaps? Narrative expert-based reviews don't really talk about that. Okay, so I've talked about a lot of problems, and therefore it was obvious that there was a need in order to create a process where you could come up with an evidence-based decision, so a decision that's solely based on the scientific evidence, where decision makers can clearly say, okay, this is what the science says, therefore this is how I need to act. And so the NAS, again, I've kind of mentioned that the National Academy of Sciences has reviewed kind of this chemical assessment process in the past. They've published several reports where they've recommended this process of conducting systematic reviews. They recommend this specifically for EPA processes, but obviously other government agencies also do chemical assessments. So they could also kind of incorporate this process into their approach. So now I'm going to talk about systematic review. So we wanted to incorporate systematic review into environmental health. And the good news is we didn't have to figure out how to do that from scratch. We actually know how to do systematic reviews, and that's because the medical community has been using systematic reviews for about 3 to 4 decades now at this point. So the medical community, if you have a new pharmaceutical or if you have a new intervention that you want to use to start treating patients, they basically do a very thorough systematic review of all the evidence and trying to figure out, is this new pharmaceutical any better than the existing pharmaceutical? And does it have any adverse side effects different from the existing pharmaceutical? And you need a sufficient body of evidence to say this new thing is better than the old thing, therefore we're going to start recommending the new thing to our patients. This whole process is overseen by the Cochrane collaboration. So you may have heard, if you're familiar with the medical literature at all, you may have heard of Cochrane reviews. These are basically evidence-based medicine techniques to transparently and systematically evaluate the medical literature. And it's really intended to prevent harm from treatment decisions. So if you're moving from one accepted pharmaceutical or intervention to a new one, you want to make sure that you're not introducing any new harms and that you're introducing at least the same level of benefits, if not greater benefits, with the new pharmaceutical or the new drug. So this is the Cochrane approach. And again, they've been developing this for about 30 to 40 years now at this point. So it's pretty highly, it's highly viewed. It's been validated. They've really empirically demonstrated that these methods work. And they've conducted thousands of different systematic reviews. The way that they define systematic review is it's a predefined multi-step process, and the goal is to identify and select, critically assess, and synthesize your evidence to answer a specific research question. So basically, it outlines that whole process of how you go from searching the evidence to making your final decision, and then deciding whether we need to act or not. That's basically what we want to do in systematic review as well, for environmental health. But there's one key difference between clinical sciences and environmental health. So both of them kind of have the implications to affect millions and millions of people, right? So things that doctors do, if they're prescribing a drug or prescribing a treatment for a disease, that has the ability to impact millions of people. In public health, it's the same thing, environmental health, right? So when EPA is making a decision about a chemical, that also has the potential to impact millions of people. But the main difference here is kind of well, there's a couple differences here. The main one is kind of thinking about when people are exposed to environmental contaminants versus when they're exposed to a pharmaceutical that their doctor prescribes them. So in the clinical field, if you're familiar with how drugs are tested, you've heard of like phase 1 and phase 2 trials. You might know that when a new drug is developed by a pharmaceutical company, it has to go through all of this rigorous testing. It has to go through cell testing, it has to go through animal testing, then it will get approved to be tested in humans, where they go through randomized controlled trials. Again, like I mentioned before, randomize 2 groups of people. You give one group of people the drug, the new drug, the other group, maybe the old drug or a placebo, and then you kind of track them over time to see what happens. And then only after it's kind of passed all of these phases of testing can doctors then routinely prescribe this pharmaceutical to their patients. So it goes through all of this rigorous testing. Now when you think about environmental chemicals, that story is really different. So chemicals in the United States are regulated under the Toxic Substances Control Act, which is an older act from 1976 that was more recently in 2016 revamped. But it still has a lot of flaws. So basically, when it was first introduced in 1976, almost all of the chemicals that were produced at that time, there was about 62,000 chemicals back then, they were basically grandfathered in and they didn't have to have any health assessments. That's why we only have that little tip of the iceberg. We only have health information for about 200 chemicals, because the rest of these, the EPA said, well, we're already using them in the marketplace, keep using them, we don't really need any health information because they're already out there. And then for new chemicals, sometimes they're assessed by EPA, but even now with the 2016 Lautenberg amendments to TSCA, we're still seeing a lot of chemicals just passing through EPA where they say, fine, use it, we don't need any more health information. So a lot of these end up going into our marketplace, into our homes, in our workplace, and people get exposures, again, without any health information. So it's very different from clinical sciences, right? Here we have all of this data first before it enters the marketplace, whereas in environmental health, it can enter the marketplace with basically no health information. And only after it's in the marketplace can we conduct epi studies, we do cells testing, we do animal testing, and we try to figure out what the health effects are. So that's the main difference between clinical sciences and environmental. And one other one is kind of this RCT, so the randomized controlled trials. We don't have RCTs in environmental health, right? I already mentioned why, mostly because of ethical reasons. So a lot of times we're kind of relying more on epi or observational studies and then animal testing. It would be a lot easier if we had RCTs, if we had RCT data available to us, because that process of randomization eliminates a lot of the confounding factors that you might be worried about when you're looking at an observational study. But unfortunately, we don't have that. Okay, so what that means, those two main differences, the RCTs and kind of when chemicals enter the marketplace, means that we couldn't take this process from Cochrane for systematic review and apply it directly to environmental health. Because number one, we don't have RCTs, and number two, a lot of the data that we rely on is animal data. So we're kind of struggling in environmental health with this problem that one of my advisors at Hopkins used to call the mouse to man problem. So we have a lot of animal data, and now more and more we have cell testing data. And we need to figure out, with all that animal testing data, how do we take those results and then figure out what it means for humans? It's not a very easy thing to do. I mean, sometimes when a mouse gets cancer, that means the human will also get cancer, but sometimes that's not the case. So how do we figure that out? How do we figure out when an animal study says something, is it going to be the same thing in humans? Okay, so our approach to this was the navigation guide. So this is our systematic review approach. I'm going to brief through these slides really quickly. I want to talk more about the application through a case study. But the main thing I wanted to highlight was this was a collaborative effort. So it wasn't just 4 academics kind of sitting in a room at UCSF, scribbling down a framework, which they decided would work best. we brought in people from government, we brought in people from both federal and state government, we brought in people from NGOs, and it was a huge collaborative effort where we all worked together and thought about what our needs were for these environmental health chemical assessments and how we could satisfy these needs through a framework for systematic review. And we also had people who were experts in the Cochrane approach, so they could say, this is how Cochrane does it, how can we modify that for an application to environmental health? So this was a 2011 paper that we published in Health Affairs, which basically outlined this navigation guide framework. These are all the lovely people who were working on the Nav Guide framework. And so you can see kind of the representation from all different academic, government, and NGO communities. And this was the ultimate framework that we ended up working with. So we had kind of 4 different steps, and within each of these steps, there's even more like sub-steps. So it is a complicated process, but this is the overall framework for how we specify the study question, select the evidence, rate the evidence and the strength of the evidence, and then grade the strength of the recommendation. Now we only kind of work with these first 3 steps because these kind of overlay with kind of how risk assessment is done. This last step is more kind of a risk management decision. This is when you have more than just the scientific information about making a decision. This is when you consider other things that you would consider in risk management, like, is there a less toxic alternative available? If there is, even though the science might say, well, it might not be that much of a concern. But if you already know there's a less toxic alternative, you would probably just select the less toxic alternative. You might want to also look at, so again, this is kind of developed from Cochrane. It's talking about patient values and preferences, but maybe you would look at societal preferences, right? One example of that is BPA. For BPA, there was a huge push from the scientific community and from the public about getting BPA out of plastic bottles and out of kids' bottles, right? So the science was not as strong for BPA. There was a lot of evidence that BPA was harmful, but there was some evidence, pretty strong evidence that BPA was not harmful. But because there was such a push from the public, the industry really felt the pressure to voluntarily take BPA out of their products. So this is an example where you might have societal values and preferences kind of overwhelm the scientific information. So I'm not going to talk about step 4, but I will focus on steps 1 through 3. And the main thing I want to say here is this systematic review process aims to be transparent and reproducible, which I've said a couple times already. But note that it really doesn't eliminate the need for expert judgment. Like when you're going through and evaluating scientific studies, you still need somebody who's an expert in the field to say this study is good or this study is bad. This study did not account for all of the confounders that I would be concerned about. Or this study had a fatal flaw in the way that it was designed, or how it recruited its participants. So you still need people who are scientific experts. It doesn't just kind of automate how you do a systematic review and evaluate the evidence. You still need those people who are in the field, who know what they're talking about, who can evaluate the evidence for you. The benefit of systematic review is it really forces each expert to transparently document the judgments they make along the way. So that means if another person is coming through and seeing your systematic review and reviewing your systematic review, they might say, well, I agree with you up until this point where you made this decision, and then you did this way. But if it were me, I would have done it this way, and then they can kind of carry that decision through. It's kind of like one of those choose your adventures, right? They can say, oh, I would have gone here instead of there, and then they could follow their decision and see where that would have taken them for their final conclusion. But systematic review allows you to do that. You can figure out where you would diverge in your kind of thinking of those two scientists arguing with one another, thinking about where they diverge and then how that might impact the final decision. So this approach is called the navigation guide, but there's a lot of other approaches out there which also kind of apply systematic review to environmental health. One approach is this, it sounds probably less fun than it is. Oh, it might not be that fun either. It's the OHAT handbook. And this is kind of exemplifies how government loves their acronyms. So this is part of the National Institutes of Health, the NIH, National Institutes of Environmental Health Science, NIEHS. It's part of the National Toxicology Program, NTP, and then within that, there's the Office of Health Assessment and Translation. So it's this little group within NIH who focuses on doing chemical assessments, and they focus primarily on hazard ID. So they developed this framework for doing systematic reviews for their purposes. It's actually really similar to the NAVGUIDE framework. Actually, one of the people who are in OHAT, he's actually the acting interim director right now, he was on that NAVGUIDE work group. So he was kind of with us from the beginning as we developed our approach, and then took that back to his office and said, okay, we're going to develop something a little similar that's targeted for our interests. EPA also has developed a systematic review framework as well. So the part of EPA that does hazard identification and dose response assessment is the Integrated Risk Information System, IRIS program. So they also have a systematic review approach up, which they follow. And again, it's a little similar to the NAVGUIDE. They all kind of have similarities and some minor differences. Okay, so this is kind of how you would conduct a systematic review. And thinking about if you had the human evidence, you kind of go through these steps. So first you develop a PICO statement, and I'll talk about what that is. And then you do a systematic search of your literature. Then from that, you identify what the relevant studies are. From those relevant studies, you extract data and you conduct data analysis. If you can, you do a meta-analysis. And then you rate the quality of all of that evidence and the strength of all of that evidence, and then you come to your final conclusion. Now, there's a lot of steps in this process, right? And we're stressing that you want to be transparent and reproducible. So how do you do that? The main way that we do that is publishing a protocol. A protocol is basically a rule book. It's kind of like if you were doing a dissertation and you have to write your research proposal and your research plan. You're basically saying, this is what I plan to do at each step. This is how I'm going to select my studies. These are the search terms I'm going to use. This is how I'm going to decide if the study is relevant or not relevant. This is the data that I'm going to extract. You're basically outlining every single step. And the thing about a protocol is it's really important to pre-publish your protocol. So that means to make it publicly available before you actually go and do any of the stuff that you said you were going to do. The way that we pre-publish our protocol is through a website called Prospero. This is run by the University of York, and it's basically a database where anybody can go in and pre-publish a protocol. The nice thing is then you have documentation. You can say, well, back in January of 2018, this was my protocol, and then now I'm publishing the paper a year later. And you can go back to my protocol and make sure everything I did in my paper matches with what I said I was going to do in my protocol. Or if there's deviation, there's got to be justification for why you deviated. The other nice thing is if you have a protocol, it can avoid wasted research resources too. There's always this fear in academia that we'll get scooped, that somebody will be working on the exact same research project as us, and then they'll publish it before we publish it. This can help avoid that, at least for systematic reviews. So if you go into this database, if you plan to do a systematic review on arsenic and cancer, you can go into this database and see if anyone has done it before, or if anybody's currently working on it. That can make sure that you're not deduplicating efforts. Okay, so this is the process for how we would evaluate the evidence. So there's a lot going on here. So there's 3 different steps here. The first step here is looking at risk of bias. So risk of bias basically says, is there anything in the way that the study was designed or implemented so that it might have biased the final results? Like for instance, did they use an exposure metric which consistently overestimates one's exposure or consistently underestimates one exposure? If they did, then you would expect that to affect the association estimate that they're reporting. There's all these other factors too, like confounding is a big one for epi studies. Did they control or adjust for all of the important confounders? Or did they leave out smoking, which you think is a really important one? All of these things you would evaluate and then assess whether it's low risk or high risk. When you evaluate risk of bias, this is actually the only step where we do it for each individual study. This whole rating then feeds in as one criteria for evaluating the quality of evidence. So we did it for each individual study. We say, Smith et al., they have these risk of bias ratings, and then Johnson et al., they have these risk of bias ratings. Okay, now taking a step back over all of my studies, are they doing pretty well, or were most of them rated high or probably high risk of bias? So if they were mostly high or probably high and you were concerned about it, you might downgrade your body of evidence. You might say, I have a lot of studies, but they're not real good. A lot of them are pretty biased. The other things you want to look at are whether it's direct evidence for the study question that you have. Or is it kind of looking at the study question but indirectly? It doesn't make that link with the exact study question that you're looking at. You want to see if there's consistency in the results that you're seeing, if there's precision, and if there's possible publication bias, which is a big issue in academia. You also want to see if there's a large magnitude of effect. So if you're worried that there might be some risk of bias and you don't think that association estimate is precise, maybe it's actually higher, maybe it's actually lower, but you actually see a large magnitude of effect, like if you see an odds ratio of 5 or 10 in environmental health epi studies, that's a big odds ratio, right? So then if you're worried about risk of bias, well, maybe it's a little bit lower than that, but you're probably still seeing overall a positive effect. So then you might actually upgrade the criteria based on that. And then you also look at if you see a dose-response relationship and if they're all possible confounders that have been accounted for. So you rate the overall quality as high, moderate, or low, and that's your final rating for quality. And then this whole thing feeds in as one consideration for strength of the evidence. So you either have high quality, moderate quality, or low quality. And then you look at the direction of the effect, the confidence in the effect, and anything else that might influence certainty. And these are your final certainty ratings. So your ultimate, remember that arrow that went all the way through and you had overall conclusions on the right? That's your final rating for your overall conclusion. Either you have sufficient evidence, you have limited evidence, inadequate evidence, or sometimes you might even have evidence of lack of toxicity. Okay, I'm not going to go through this one in as much detail. It's basically the same thing. You look at risk of bias, but since it's an animal study instead of humans, you might want to look at whether or not the sequence generation was random or not, basically how the animals were randomized to the study groups. But same thing, you look at risk of bias, it feeds in as one element of quality. You look at the same downgrade criteria here. This all feeds into quality. You look at the same strength considerations and come up with similar ultimate ratings for your animal evidence. And then this is how you integrate the two together. So now you've rated the animal evidence, you've rated the human evidence. You bring these together, and up here, this is how you rated the animal evidence. This is how you rated the human evidence. And you basically just see like where these things converge and then what your overall conclusion is. So if you have sufficient human evidence, maybe you have inadequate animal evidence, but that's okay, because since you have sufficient human, you would still say that it's known to be toxic to human reproduction. Basically, the human evidence trumps it. Whereas if you have limited animal evidence and limited human evidence, you would end up as possibly toxic, because neither of those were strong enough to convince you that you had sufficient or probably toxic evidence. Okay, so we have conducted to date six different case studies applying this approach. This is not an easy approach, so I breezed over it. If you don't fully understand it, that's completely understandable, because this was our very first case study, this PFOA and birth weight case study. We did human evidence, animal evidence, and then the integration of the final evidence. And we published this in 4 separate papers, looking at, one was an overview, one was human evidence only, one was animal evidence, and one was integration of the evidence. So it's a complicated enough process that we needed one paper for each thing. So if you don't fully understand it, that's probably why. There's a lot of moving pieces behind each of these case studies. This was our first case study. We've published all of these except this last one, formaldehyde and asthma. That one is currently under review at +1, but we published all of the other 5 so far. Okay, so now I'm going to talk a little bit about mechanistic data. So you'll notice before, when I was going through that whole framework, I really only talked about human evidence and animal evidence, right? But you're missing kind of a big piece of the puzzle now, especially kind of nowadays, in more recent times. So nowadays, we're starting to see more data that's being generated basically in a petri dish or a test tube. So this is called in vitro data, where we're looking at cells or other things that can be grown in a petri dish, either human cells or animal cells. And you can actually expose these cells to chemicals and see how they impact either cell reproduction, how it impacts cell death. You can look at different measuring techniques for activity within these cells. And you can try to make inferences for humans and animals based on this. So this is huge. I mean, this is getting a lot of influx of money from EPA, FDA, other federal government agencies, A lot of academic researchers now are testing in cells. They're doing more advanced things like this is called an organ on a chip, where you actually have different cellular processes that kind of make up one small piece of what an organ does, like a liver. There is a researcher up at Johns Hopkins University, Thomas Hartung, and he has these what he calls mini brains. It's basically these like little cellular replications, not exactly of an entire brain, but of like key neurological functions that you would see in a brain. And he can expose these mini brains to chemicals and then see how it might impact neurodevelopment. So it's pretty cool what is coming out. And the other thing that's happening now is more of this is getting automated. So you can have these machines who are able to like get petri dishes, load them up on these 36 well plates with cells, expose them to chemicals, put them back, and you can have a machine that basically does this all day, and it can do it about 50 times faster than a human being would. If you've ever worked in a lab, which I did my first couple of years when I was an undergrad, and you've like pipetted these petri dishes or test tubes for like hours, and your thumb gets like really stronger than the rest of your 9 fingers, and it's really awkward, you know that this is a lot of work. So the fact that we can use machines to do this has basically expedited this process exponentially. So what does that mean? That means we're getting a lot of data from in vitro systems, but we don't really know how to analyze this. So this in vitro data has a lot of benefits over the traditional way that we've done things. So usually for animal studies, we have to test at really high doses. That's because we want to see an effect. But it's also a high dose that we know that humans would not be exposed to. But for in vitro data, you can test at any dose you want. So you can test at a bunch of low doses, you can do medium, you can do high doses. Because you're only doing these 96 well plates, it's really easy to have a wide range of doses. But when you have animals, I mean, animals are really expensive. You're tracking them for over 2 years usually. So if you want to do 5 or 6 different doses, that gets really expensive and really unwieldy. So usually they just do high doses for animals. But that's the benefit of in vitro data. For animal data, you also usually look at apical endpoints, things like cancer, things like liver disease. But for mechanistic data or cell data, you can look at kind of earlier markers of toxicity pathways. You can look at inflammation. You can look at these minor perturbations of cellular pathways. You could do that for an animal study again, but it would be very expensive. So usually they look at just kind of apical endpoints at the very end, indicators of disease. But here for in vitro, we have a lot more flexibility with what kinds of outcomes we can look at. The other benefit to in vitro data is you can use human cells. So these mini brains, these mini livers, all of these are usually human cell based. So you don't have to do that animal to human extrapolation anymore. You're looking at humans to humans. Okay, that's what I want to say about that. So this is starting to be very promising. And again, the NAS had come out with a report, I forget when this was published, maybe 2014, or no, this was 2007, it was longer ago, where they actually talked about the promise of these methods and really encouraged researchers to start looking into developing these data and starting to incorporate them. I'm going to skip this slide real quick. I'm going to talk about this one a little bit more. So this is kind of how the EPA and other folks are thinking about incorporating this data into risk assessment. This is called an AOP, so it's an adverse outcome pathway. Basically what this is a pathway that proposes how a chemical might interact with one's internal body on the inside and basically kind of have this cascade event that ultimately leads to an apical endpoint. So you know if you're exposed to an outcome, usually it leads to a disease. But how does it get to that disease? So this is a proposal of how you might have a chemical interacting with a cell on the molecular level, and then kind of causing this cascading effect down by cells and by up to the tissue and the organ level, up to finally the adverse outcome. So this is kind of like mapping what you think happens internally in your body in a cellular fashion in order to get from exposure to the chemical to the disease. So this is called the molecular initiating event. This is kind of like the one hit where the chemical starts this cascade of events. And then once this gets disturbed, it has these key event relationships, these key ERs and KERs, and it causes this cascade of events until you ultimately get to that final adverse outcome. So this is an AOP. This is getting really popular with scientists now, where they're trying to map everything. They're trying to figure out how does exposure to a chemical ultimately lead to a disease? What are all the cellular and molecular processes that cause that? So this is where a lot of that in vitro data is coming into play. You might take one of those mini brains and you might perturb it and then see what are some of these key events and how are these two things related. So, as I mentioned before, we have a process in systematic review for human and animal data. But we don't really know how to do in vitro data. So one of the problems is nobody's really done this for in vitro data. So nobody's thought about how to evaluate the internal validity of a mechanistic study. So if you have a study that's done in the mini brain, how do you evaluate that quality? How do you say these are things that might bias the results because of issues with how the study was designed or implemented? We don't have a risk of bias approach. We don't have any established frameworks for how you rate the quality or the strength of this type of evidence. There are some approaches which have tried to do this. So this is kind of a checklist of different study quality criteria, some which are kind of varying quality. You'll see the navigation guide here. We look at human and animal studies, but we don't look at in vitro studies. You'll see there are only a handful of studies that do look at in vitro studies. This would be the ARRIVE guidelines, the Clinish system. OECD has a guidance document that looks at this, and then there's a tool called the ToxR tool, which proposes different ways to evaluate this. But none of these have really been validated or tested. But there are, the point of this slide is to show you there are some frameworks that start to propose how this might be done. So there's kind of a path forward in thinking about, okay, how do we start incorporating in vitro data here? Some of you may have heard of the Bradford Hill considerations for epidemiology studies. There are some scientists that have tried to take these Bradford Hill considerations and modify them for an approach to in vitro studies. So they've taken kind of 3 key things that are related to, that have been proposed originally by Bradford Hill, and thought about how you can apply these for evaluating an in vitro study. So that's looking at biological plausibility, essentiality, basically whether something is essential in order to create that cascading of events, and then whether there's empirical support. There are organizations who are working on this, so this is kind of like the promise of the future. I just was at a conference 2 weeks ago where a bunch of folks from these organizations all got together and they were thinking about how to develop these types of frameworks for evaluating in vitro studies. So how might this be done? So in vitro studies might be incorporated in a similar way once you have that kind of validated framework for rating the quality and the strength of the evidence. And then in terms of how you would integrate it together, it's unclear. So whether you would have like a 3-dimensional kind of intersection of evidence where you also incorporate in vitro data, or some people have suggested maybe we just take the in vitro data and basically if it supports the human evidence, like if the in vitro data says the same thing as the human evidence, we would use it to upgrade the human evidence, or vice versa with the animal. If it supports it, it would upgrade it, but if the in vitro data shows something different than the animal or human data, you would actually downgrade it. So that's another proposal. I don't know if I have time to talk about this, so I will just mention there's another framework that I'm actually working with in one of my projects right now, which is looking at this approach to developing key characteristics. So basically this was convened by IARC, the International Agency on Research on Cancer, where they tried to develop 10 things that you would want to see in something that causes cancer, or something that causes either female reproduction health effects or male reproduction health effects. And these are some of the 10 things. So this is the example for male reproduction. So these are the 10 things you would want to see evidence for, like altering production levels of reproductive hormones. If you see that, then this gives you increased evidence that something might be a male reproductive toxicant. And the same thing for female reproduction. I'm just going to skip over this and go right to conclusions. So in conclusion, this has been a really challenging topic. I've been working on this now for, gosh, 8 years now, and we've been pretty successful. We've gotten case studies that we've published, but there's still a lot of work that has to be done. Like, we haven't even touched in vitro data yet. We're just starting to incorporate this in. So there's a lot of questions because we see this paradigm shift and thinking about generating more in vitro data. We have to really start thinking about how do we incorporate this into decision making. I don't know if any of you are familiar with this, but the EPA has just recently announced that they're proposing basically getting rid of all animal testing within the next 5 to 10 years, which might sound like a good thing, right? You probably saw the cute animals that are on my slides, and you're like, well, you know, all of these animals are being sacrificed in order to figure out whether something's harmful to humans. Wouldn't it be a lot better if we just didn't have to test in animals? Well, the problem with that is then how do we figure out if something's harmful for human health? How do we ban things, right? Do we just let humans continue being the guinea pigs when we use chemicals out in the marketplace and then wait to see if there's an epi study that shows it's harmful? If we get rid of all animal evidence, we basically have epi and we have in vitro data. And that can be a really challenging set of data in order to actually make decisions on. So I think this is going to start getting to be more and more important though, because we are seeing more in vitro data. And that's basically it. So thank you all for your time. I really appreciate the opportunity to be here. I have my contact information here. If anybody has any follow-up questions, I'd be happy to talk more, either about systematic review or my research, or we are also hiring in my department for tenure track faculty members, so always trying to recruit people. If you are interested or if you know of people who are looking for jobs now, feel free to share my contact information.